I also asked Frank, our IT Frank, to stand by just in case. Yeah. So you're gonna have a recording starting from beginning, right? As soon as you start.
It's already started. We can always add it to.
Okay, that's great. Yeah. So in this case, you guys, good luck with the whole thing. I know you spent so much time. Thank you, Rosa. So we'll, yeah, I'll be standing by in case there are some pricing or technical questions I can address. Yeah. Okay, I'll start here. Good luck.
Thank you.
Can you hear me?
I can hear you just fine, Rosa, but I have not seen your. Oh, I can see you just fine. Yeah.
Yeah, I can hear and see both of you. So.
Yeah, but the problem is I cannot use my phone.
You cannot use.
If I have a phone, you guys don't hear me.
Oh, I now can hear you. I think if you can just present that way, it should be fine.
Okay, then I will do this. Okay.
Yeah, I think I can hear you quite clearly. Hi, everyone. Good morning. Well, people are still logging in, so let's give a couple more minutes. Thank you for your patience. We'll get started shortly.
Yeah.
Okay, all right. So good morning. Let's get started. First, I just want to make sure everybody can hear me okay. From the panelists and folks online, if you cannot hear me, please, you can unmute, let me know. All right, so I assume everybody can hear me fine, so let's get started. Good morning, everyone. Really a warm welcome to today's Newomics and Thermo joint webinar. My name is Nara. I'm Newomics Senior Director of Marketing. So today I'm with my Newomics colleagues and the Thermo team are very excited to have all of you join us today. Well, as you can see here on this slide, I'll go through first today's agenda very briefly. So we will start with a brief introduction of the Newomics microflow nanospray, ESI-MS, MnESI-MS platform by myself representing the Newomics team. After that, Dr. Rosa Weiner from Thermo Fisher Scientific will share with us her story of interfacing Newomics MnESI-MS platform with the Thermo Orbitrap mass spectrometer for analysis of native protein complexes. We will wrap up today's program with a 15-minute Q&A and discussion section after the presentation. So before we present today, I would like to take a moment to introduce our speaker, Dr. Rosa Weiner. So Dr. Weiner is the Senior Manager of Omics Vertical Marketing from Thermo Fisher Scientific in San Jose, California. She joined Thermo in 2004 and is involved in all areas of genomics research with specific focus on relative quantification, DM characterization, and integrated structural biology workflows. Dr. Weiner serves as a liaison between life science mass spectrometry and Pierce Protein Research Business Unit at Thermo to provide complete solutions and workflows to customers. Rosa holds a PhD in biochemistry and biophysics from the Moscow State University of Russia. All right, so let's welcome Rosa. Also with us today is the Newomics team on the panelists, including our CEO, Dr. Daojun Wang, and my colleagues from Newomics R&D, marketing, sales, and support divisions. So the Newomics team will join the discussion after the presentation, and we'll be delighted to answer your questions together with Dr. Weiner. So a couple of housekeeping items before we get to the presentation. So you will be muted during the next 45 minutes of the presentation. While in the Q&A section afterwards, we certainly highly encourage you to unmute yourself and ask the questions. You're also very welcome to type your questions anytime during the presentation in the Q&A box, which you should be able to find on the right side of the Fec portal. We'll also make sure to address all your questions when you type in the Q&A box afterwards. All right, let me make sure muted. So let's get started. All right, so we, the Newomics team, together with Thermo, were thrilled to share that Newomics recently, actually last week, launched an innovative microflow nanospray HESI-MS MnESI-MS platform, co-marketing with Thermo. So the MnESI-MS platform features the new MnESI ion source that empowers the M3 multi-nozzle emitters to deliver significantly improved Lcms performance. The new MnESI source is designed to meet mass spec users, your needs, mass spec users' needs by offering superior sensitivity and robustness for high throughput analysis of proteins, peptides, nucleic acid, and lipids from complex matrices. So today, Rosa and I would like to focus and describe how the MnESI platform seamlessly interfaces with Thermo's mass spectrometers to address several key pain points in analyzing native proteins. So we all know that nanoflow Lcms is routinely used to achieve high sensitivity in analyzing proteins, peptides in small volumes of biological samples. However, researchers are facing certain challenges in their nanoflow method. For example, assay robustness is a constant concern, and sample throughput is often not very ideal. Where on the other hand, high throughput method achieves good throughput and robustness, but its sensitivity is insufficient in some cases. For example, clinical research when sample amount could be very limited. So further enhancement in sensitivity is highly desirable in high flow. Well, microflow Lcms with a flow rate from 1 to 50 microliter per minute could potentially breach this gap because of its balanced sensitivity and robustness. Also, we know that single nozzle emitters have been utilized for conventional current Lcms methods, including we all know that nanotip, picotip, nanomade for nano-Lcms, and standard ESI single nozzle sprayer for high flow Lcms. Well, the Newomics multi-nozzle emitter technology and the microflow nanospray ESI-MS product are designed to help address challenges from the conventional nanoflow or high flow method using a single nozzle emitter. This is how it works. So the microfabricated monolithic multi-nozzle M3 emitters split the incoming microflow eluents evenly into multiple nanoflows at each nozzle, therefore significantly enhancing the electrospray ionization efficiency and reducing the matrix effect for mass spec analysis. So to better understand this theory or this mechanism behind the M3 technology, you can imagine this is like the water flow being split evenly through a shower head, generating smaller and finer droplets. So the flow splitting capability of the M3 emitters and the resulting smaller droplets can greatly facilitate the dissolution process during the ionization, therefore getting higher ionization efficiency. Well, this year, like I have just mentioned in the beginning, we are very excited to introduce our new MnESI ion source. In fact, just this month, last week, we had the announcement. So you can see the design picture here. The MnESI ion source empowers M3 multi-nozzle emitter to deliver that improved performance. So in the MnESI source, the M3 emitter is mounted on a push-pull stage, which allows for a very easy setup of the maintenance. The fixed spray angle here can help reduce contamination. Additionally, the MnESI source is equipped with on the left you can see a high quality imaging system, a camera, to allow clear visualization of the electrospray process, which together with the clear glass window in the front can facilitate that fine-tuning, accessibility, the fine-tuning of the source conditions. It is noteworthy that the new source, it is designed in a closed format, which is to ensure your operation safety and hygiene. So the enhanced ionization efficiency delivered by the M3 emitter, as I described in the previous slides, translate into improved peak area response, higher sensitivity, and a lower limits of detection and quantitation in the ESI-MS analysis. The multi-nozzle emitter not only actually reduced the size of the droplets, improved the ionization efficiency in the ion source, it also helped mitigate the risk of clogging from protein precipitation and accumulation, which leads to greater robustness and reproducibility. With that improved sensitivity, it is now possible to analyze samples with a smaller injection amount for the dilutions, which also in a way helps to reduce the high salt contents or biological samples being introduced to the mass spectrometer. Therefore, it minimizes the contamination, helps to reduce the downtime due to cleaning, and ultimately could improve the method robustness. Moreover, LCMS method gradient could be reasonably reduced because of the increased sensitivity, which resulting in a faster runtime and higher sample throughput. Well, last but not least, certainly, the MnESI source is very easy to install, straightforward to use, which enables a real plug-and-play emitter operation for your laboratory. So the new MnESI source delivers optimal performance in the microflow range of 1 to 50 microliter per minute, while it also supports the nanoflow method from 20 to 1,000 nanoliter per minute. The MnESI source is fully compatible with all Thermo Fisher Scientific mass spectrometers. So we have designed these two models, MnESI NG and MnESI Legacy, to meet the needs of a different thermal instrument in your laboratory. On the left, the model IST01 interfaces with Thermo's newer generation mass spectrometers, such as the Orbitrap Fusion Lumos, Eclipse, Explorers, and the TSQ are newer, the TSQ newer Altis triple quad. While the model IST02 on the right works well with Thermo's legacy mass spectrometers. For example, the very popular Orbitrap 2 Exactive mass spectrometers, I believe many of you have them in your laboratory. All right. So Newomics microflow nano spray ESI-MS platform, including the MnESI ion source and the M3 multi-nozzle emitters, have been successfully utilized to enhance the assay performance and streamline analytical workflows in many key application areas, including native protein analysis, which we'll talk about today, bottom-up proteomics, targeted proteomics, lipidomics, as well as analysis of a nucleic acid in negative mode. We're also very happy and thrilled to share that Newomics now have more than 80 customers, including leading pharmaceutical companies and academic institutes who have adopted the multi-nozzle emitter technology and successfully employed the MnESI-MS platform in their research and development. Very recently, I want to mention and share this. Newomics M3 multi-nozzle emitters were featured in 2 publications from Regeneron and University of Lausanne. Regeneron published very impressive work in JASMS, and they described the development of an integrated Lcms platform for sensitive and robust native protein drug characterization, which is enabled by the Newomics multi-nozzle M3 emitter. In particular, this developed platform can tolerate a wide range of LC flow rates and high salt concentrations, which is critical for accommodating different native LC method, LC chromatography conditions. Well, on the right, you see University of Lausanne's publication, a very exciting article in Talenta. This work represents a new LCMS workflow for determining native peptides in plasma at the very low picomolar levels for clinical diagnosis. Also, it's enabled by Newomics multi-nozzle M3 emitter. So the M3 emitter attains an average in this work sensitivity gain of 25 times for peptides in plasma extract. All right, so among many successful applications, Rosa and I will share with you today how the Newomics MnESI platform interfaces with Thermo's Orbitrap mass spec to achieve that highly sensitive and robust analysis of native antibodies and a large protein complex. This will be described by Rosa. So I'll share some results on the native antibodies. We know that recently non-denaturing size exclusion chromatography, SEC separation, with native MS detection has gained really increased interest in measuring mass of complex biologics and their heterogeneities. However, conventional or currently used analytical high flow SEC LCMS method may not be sensitive enough to detect proteins with low concentrations or analyze diluted samples. So to address this challenge, we have established a highly sensitive MnESI-MS method with the MnESI source and the M3 emitter coupled with the Thermo Orbitrap Q-exhaustive plot for characterization of the native antibodies. So the NIST SRM, standard reference material, monoclonal antibody IG, was used in this study. SEC chromatography was performed using Ultimate 3000 RSLC Nano and using an isocratic 200 millimolar ammonium acetate. Performance of the MnESI source with the M3 multi-nozzle emitter and microflow of 5 microliters per minute was evaluated and compared to that of an IMAX source with a single nozzle HESI high flow needle at analytical or high flow of 200 microliter per minute. So the most abundant 5 proteoform or glycoforms of the IG were quantified to compare the performance of the different emitters. So let's first take a look at how the sensitivity gain works using the M3 emitter. So as you can see, the MnESI source with M3 multi-nozzle emitter and microflow delivered a significantly increased MS signal intensity compared to the analytical flow method using the IMAX source with HESI single nozzle needle. More specifically, we see a 20 to 50 times of improvement in peak area intensities for the native IG, as you can see demonstrated here with 50 nanogram and 200 nanograms of Ig on column. Besides comparison to high flow, we also evaluated the sensitivity performance of the M3 emitter, multi-nozzle emitter versus different single nozzle emitters, including the Thermo Fisher stainless steel single nozzle emitter, which is called a nanotech, also the new objective, picotech, and a HESI low flow needle. So as you see here, a 2 to 5 folds of gain in peak area intensity was observed for the M3 multi-nozzle emitter versus the 3 single nozzle emitters using exactly the same LCMS condition with that microflow rate of 5 microliter per minute. This gain is purely by the switching from the single nozzle to the multi-nozzle emitter. So the sensitivity gain, clearly, obviously, that higher sensitivity will lead to a lower limit of detection. So we'll see the LOD, low LOD level is below 10 nanogram of native Ig on column, which also potentially enable a wider dynamic range for quantitative measurements. So we all know that higher sensitivity, better dynamic range achieved here could allow for a more confident detection and ID for low abundance proteins and variants. Also with that sensitivity improvement, it is possible to analyze samples with smaller amounts, with postal column splitting, for example, further dilution, shorter gradients for higher sample throughput. And as discussed before, when we use smaller sample amounts and further dilutions, it will help improve the method robustness by introducing less sample into the mass spectrometer. So we all know that robustness is without question another major consideration when developing a CMS method in the laboratory for routine applications. In particular, native LCMS method, because it's continuously introducing the aqueous bulk solutions to the analyzation source, leading to certain challenges in maintaining the spray stability and the signal intensity over extended analysis time. So in this experiment, we assessed the robustness of the MnESI-MS platform for native Ig with 150 consecutive injections and almost over 2 days of a continuous operation. So you can see a stable peak area response observed over that 150 injections and 2 days, and the CV of the ratio of each of the glycoforms here quantified range from 4 with the more abundant ones to the less abundant ones, even have 15%, with an average of 7% over that 2 days of analysis time, which really indicating an excellent robustness and reproducibility from the M3 emitter and the MnESI-MS platform. I would like to also share our customer Regeneron in their recent JASMAS publication evaluated the robustness of their native LCMS platform developed using the Newomics M3 emitter FRAC. So this is very clearly demonstrated with that over 50 injections, and they have validated for over 24 hours. They see a very stable absolute signal intensity over that long analysis time, with the CV value reviewed here 4% or 6% for native monoclonal antibody analysis. Right. Moreover, in this Ig study, we observed the charge envelope of Ig from the M3 multi-nozzle emitter shifted towards a higher MOVZ range compared to data from corresponding the most abundant charge states. You see the shifting from the HESI high flow, the 29 plus to 28 plus in the HESI microflow, while to 27 plus is a much lower charge state and a higher MOVZ in using the M3 emitter. So this suggested a better preservation of the native conformation using the multi-nozzle Newomics M3 emitter. So we speculate this microflow nanospray function of the M3 emitter might result in a more softer ionization environment for Ig. This observation, in fact, we believe is quite significant because maintaining protein native state, it is really important and critical in studying protein target interactions and non-covalent complexes in gas phase. In conclusion, so we have I've showed you the Newomics microflow nanospray MnESI source and M3 emitter interface with the Thermo mass spectrometer Orbitrap QX Plus to streamline analysis of native antibodies. The MnESI platform achieved really improved the sensitivity, great robustness, which allows for smaller sample amounts, enables higher throughput, and provides better preservation of the native protein matrix state during the ESI analyzation process. Okay, so thank you for your time and attention. This is pretty much the end of my presentation. If you're interested in Newomics MnESI-MS products and solutions, please feel free to check out the new product page here, where you can find a very informative collateral and the literature, including brochure, flyers, product spec sheets, product video, application notes, and also recent posters and link to customer publications. So finally, I would like to take a moment before Rosa's presentation to really acknowledge the Newomics team and the Thermo team. So the new MnESI platform was brought to you, the mass spec users, via a very great team effort with contributions from many hardworking people here in multiple functional areas and divisions from the two companies. Thermo and Newomics first joined forces in 2019 for the commercialization of the M3 emitter, and today we are more than happy to expand our successful collaboration and co-marketing activities towards the new MnESI-MS platform and solutions. And the ultimate goal, obviously, is to best meet your requirement needs from your research and development. All right, that's all from me. Thank you. So now I'm going to to Rosa.
Okay. Hello, everyone. Now, thank you for the nice presentation. Can you hear me well?
I can hear you very well.
Okay. Thank you again. Yes. And hello, everyone. Today, I will be present our new workflow using the new Newomics source for native MS, one of the mass spec technique we use for the structural biology. Structural biology plays a major role in drug design. All information obtained by functional proteomics is necessary to diagnose and design disease-specific drugs, starting all the way from the genes to the protein structure. Structural biology determines the protein 3D structure at the atomic level. There are several main techniques used to determine structure of the biomolecules, such as X-ray, NMR, and the cryo-EM, as well as the mass spectrometry, which usually plays a complementary role. The native MS is one of such a technique which we're using for as a complementary in the for the structure for the solve the protein structures. It's based on electrospray ionization, whereas the biological analytes are sprayed from the non-denaturing solvents at the near neutral pH. And because of this, it's able to preserve native-like structure in the gas phase and the mass spectrometers. It can be used for the wide range of the different applications, such as the drug, ligand, protein interactions. It can be studied nanomachinery, such as the proteasome and ribosomes. It also can study the whole virus assemblies. But it also has some challenges, which are outlined here, and we're trying to overcome with the new development. As always, on the Thermo Fisher, we have complete workflows for each our application, and we also starting from the protein introduction, then MS analysis, and the data analysis. By the way, we just introduced our newest version of the Biopharma Finder 4.0, which we use for this such analysis. The mass spectrometers, which use for the native MS, those have some special requirements. And the most instruments we're using for this workflow, it's our flagship instrument, Orbitrap Eclipse, which we introduced last year. And it's our first hybrid, which has the extensive capabilities for the native omics. It's have a mass range up to 8,000. It also have a different pressure regimes necessary to study the native proteins in native protein complexes. And also in our flagship instrument we're using for this type of analysis is Q-Exactive UHMR, which we introduced in 2018. This is only one instrument which has a mass range up to 80,000. It also have a capabilities to isolate ions up to 25,000, and it's also does not have so many digitization techniques, of course, as Eclipse, but it does have 3 different techniques which we can use for the top-down analysis. For these particular studies, we actually used Q-Exactive UHMR. As I say, that's our dedicated instrument for this type of analysis. When we're talking what is important for mass spectrometer for this type of analysis, we always think it's supposed to have a very good dissolution capabilities, as well as the high mass resolution and able to transfer the large ions. So this particular instrument, UHMR, has 3 ways how we can do efficient ion desolvation. We can do, of course, the traditional in-source CID, the yellow region. We also have extended trapping, HCD, but we also have a unique feature specific for this instrument. It's the in-source trapping, and it's the area between exit lenses of the shape and to the injection flat pole entrance. In this case, we actually can either pass ions through, or we can trap, apply the energy, and also perform the desolvation to remove all this non-specific solvent adducts, the detergent. We actually use this approach especially for analysis of the membrane protein. So we do have this workflow. However, native mass spectrometry was around for some time, and as I show, like new instruments are available, but still not many labs are using. So what is the reason? So we feel like there is 2 main barriers for adoption. This workflow is very manual. So the first step when we started, we do the buffer exchange. So native MS is intolerant to non-volatile salts. So we need to change whatever the buffer is to the volatile salts as ammonium acetate. The second barrier for adoption, we feel it's how the sample is actually introduced to the mass spectrometer. For this, we're using the metal coating emitters. And this is actually required some special techniques. So these emitters need to be clipped, as you can see on this video. And reproducibility for non-expert will be low. And it takes some time, it's delicate. And all these 2 very manual steps, we feel it's really prevent for the wider adoption of such technique. So that's why we always looking, for the new capabilities and how we can improve and usability of this workflow. So that's why we partnered with Newomics to elevate the new source exactly for this workflow. In our evaluation, we use 3 setups. So the 2 classical setups, of course, the gold standard for the native MS is the static nanospray, and also we use just the electrospray Lcms setup, as well as setup using the MnESI source. And you can see actual picture of this source on my UHMR instrument. In both cases, when we use the electrospray source, HESI or MnESI, we use the liquid chromatography, we use our Flex Vanquish, and we use our size exclusion pack column, which was 2.1 by 15 centimeter. And the flow rate at the 50 microliter per minute. And in the case of the MnESI source, because it's a microflow source, we use split flow. So in our split flow was 1 to 10. So 5 microliters went to the instrument and 45 microliters went to the waste. And so this was our setup. And the first experiments, we basically benchmark performance of this new setup for the GRUEL. And the GRUEL is a 40-mer complex. It's basically the system suitability standard or a very standard protein, which we always check the instrument performance for the native MS. So in this case, you can see we detected the GRUEL easily using all different setups. However, the signal to noise was very different for using the HESI source versus the Newomics source, and you also we can see the signal, and also we were very pleased when for the signal to noise with Newomics source was as good, almost as good as with the classical static nanospray. And the charge envelope was also looks very similar between these two setups. And also we were actually surprised we did even detected the 15-mer in this case, maybe because we used an LC, and the peak LC peak was quite the LC is actually improve our signal to noise, and we detected this 15-mer, and in this case, because we using our preparation GRUEL is kind of active and it's trapped the extra molecules. It's also very interesting. So here we actually, for the LC experiments, we injected the same amount, about 6 micromolar in both cases, but in this, for the Newomics source, we use only 1/10 of it. And so the amount we loaded or we analyzed in case of the Newomics source was basically the same we will be using for the static nanospray. So for this benchmarking experiments, we were pleased with the results. And I also say we didn't actually do much optimization on instrument side. We just use our GRUEL default tune method, which comes with the UHMR. So encouraged by these results, we moved for something more challenging complexes, which is the rabbit 20S proteasome. This complexes consist of the 28 subunits, 14 subunits, alpha subunits, and 14 beta subunits. And it's one of the important complexes. Proteasome, it's about 700 kilodalton. And what is known about this particular complex is actually not that stable as a GRUEL. And especially we're using the rabbit band, which is in a recent publication, for example, from Michael Sharon lab. She really say that's one of the less stable 20S proteasome complex. So for us, it was very interesting if we can actually detect the native complex using this new source. And you can see the experiment. So we actually, when we use our just default GRUEL tune, we can detect intact already complex, but we also see a lot of dissociated subunits, a lot of alpha dissociated subunits. And our default method is including the S lenses about 200, which we use to improve the transmission of the large ions. And what we did, so we actually changed, if we change the S lenses to 0, in this case, we were successfully actually detected duct complex without very negligible, basically no any subunit dissociation. So we were very pleased with results, and you can see, yes, our envelope, the charge envelope was a little bit wider than we have as the static nanospray. However, the data were very compatible. And we were pleased. So we have really this workflow we now extending with a different application. It's not something very stable, but also as labile as a 20S proteasome. Because I work in a group which is doing the structural biology, we communicate a lot with our colleague from the division which is manufacturing the cryo-electron microscope. So as you know, we acquired the FEI a few years back, and since that we really close to work on this group. And if you're talking about the cryo-EM workflows, this workflow is really starting from, again, sample to the structure. And we're always looking for ways how we accelerate this process. To have a very high resolution structure, you need to start from the very good quality of the sample. And this is actually common there, like they call the frustration loop 1 or frustration loop 2. And we were thinking how we can actually accelerate this process and how we can, improve this or remove this bottleneck on the frustration loop 1. How we can do the sample screening using the native MS. In this case, so we actually use this setup, and after we optimize using the GrowL 20th proteasome, we actually perform this experiment for the sample screening. In this case, we were screening again 20th proteasome, but not from the rabbit, from the bacterial one. It's a Thermoplasma acidophilum, or a T20S. In this case, you can see what we did. So the samples were prepared in 3 different solutions, and they were analyzed by both the native MS and the cryo-EM. As you can see here, it was done completely in automated fashion. We didn't do buffer exchange. We don't need any emitters. It was automated. So what we can see, the best results, which we get by cryo-EM, very homogeneous samples. It was actually the best results we get from native MS too. So it was most homogeneous. So we didn't see aggregates or fragments. The worst conditions, which were for get in pH 9, were both detected by the cryo-EM. There was multiple aggregates and very few good particles. Exactly the same. We also observed. We see a lot of aggregation and the fragments. So again, we were pleased with the results, and we were obtained this. This screening didn't take us long time. It was again completely automated without any users interactions. So and now we're extending more because we're doing a lot of sample screening before cryo-EM. And then the conclusion. I think we believe what the new platform significantly improved usability aspect of the native MS using microflow Lcms setup. We were able to achieve comparable data quality as with conventional static nanospray, but with all the pluses from automation, higher sample throughput, and higher reproducibility and robustness. And we also show what the native Lcms can be used as a high throughput method for cryo-EM sample screening. Then I like to acknowledge all my colleagues from the Thermo who is actually built this Q-Exactive UHMR instrument, also my other colleagues, which we developed this workflow. Of course, my collaborators, which we use samples from this study, and of course the Newomics, especially Pan, who help us to set up those. And thank you very much for your attention.
Okay, thank you, Rosa, very much for that very nice presentation. Okay, everyone, now it is the question and answer time. So you can please feel free to unmute yourself and ask your question. Yep. Actually, I do have a question here, several from the Q&A box. I guess, Rosa, there's several questions for you, so I'm going to start with those questions first. Yeah. Also, in the meantime, we go one by one, and you can also ask questions by unmuting yourself. So, Rosa, the first question is that do you have any idea of why a higher charge state distribution was observed for GrowL using a nano capillary?
I think it's you kind of also answer, right, in your presentation. We feel it's the conditions when we introduce the ions through the MnESI source, right, is quite soft. And it's probably helping us to preserve more native state. Yeah, because we're not doing any gases, right, for this, and no cooling. Yeah. You know, that's my
Right.
that will be my explanation. Yeah.
Thank you,
And it's
Rosa.
also and it's also depending a little bit from the different complexes, right? Yeah,
Right.
and also we always using the 200 millimolar ammonium acetate, which we're not always using, for example, for static nanospray experiments. But for this workflow, we found that we really need to use the higher concentration of the ammonium acetate to have a better charge envelope.
Yeah. Thank you, Rosa. Just to add a little bit, I think what we think is that the software analyzation environment created by because we don't necessarily have to use gas by the MnESI source and M3 emitter is pretty much creating just similar to the NanoFlow or NanoSpray environment, which will have a more of an efficient ionization software to preserve the native stage. At the meantime, also will because it increases ionization efficiency. So it also depends on the proteins we study and the complex we study. Hopefully that answers that question. Okay. All right. So the next question is I think that's for me. Is the multi-nozzle emitter compatible with instruments from other vendors? I think to answer that question, I think we have our marketing agreement signed with Thermo. We're working very closely with Thermo. So the first generation of the MnESI source release by the end of this month is going to be the shipping release. It's going to be the 2 models that I described for thermal mass spectrometers. We were working also with other mass spec vendors. There will be another model available for glucose mass spectrometers in early next year. As for other vendors, we're working closely with them. Hopefully, we'll be able to bring this solution and the technology to all your instruments and different platforms in your laboratory in the near future. Yeah. All right. Another question here. Sorry, I feel like I'm muting. What I can do is to see if I can get all this. I'm muted. The background might be a little bit more. So if you can individually muting yourself, except you're asking questions, that's great. Okay. So the next.
Actually, I also can respond. Yeah. So actually, we see mostly like low charge states. So I'm sorry, I was confused. Now I see the questions. No, when we use on this platform, actually, we see the low charge states for both the GROEL, right, and for the protozoan, it's actually a little bit depends how the protozoan were prepared. But in general, we actually see low charge states. We see very compatible with our static nanospray. With the HESI source, as also now is telling, we actually see much more high charge states. So you show right for the antibody, and we actually observe the same for the bigger complexes as well. Yeah, we actually very pleased. We really see the low charge state. Yeah. So
I agree
because only right now I can actually see the question, so I didn't see it. Okay.
Right.
Yeah. So
to be very clear, I think Rosa, you also presented the comparison on the high flow, which would see very similar trending. So you will see a lower charge state, which, once again, I think what we can comment is that with the M3 multi-nozzle emitter, you will generate more soft and close to nanospray environments that will help preserve the native state. Okay. The next question I have is, you both tried membrane proteins with bound lipids. Can you get away with running only a few microliters? Can you lower the spray voltage to 1.1 kilovolts, as well as the capillary temperature to retain the non-covalent interactions? This is actually a great question. So I think, Rosa, please, you can feel free to comment. I think as for now, we have just launched this new source product. We have worked on native antibodies and native complex, protein complex. as Rosa presented to you. I know that membrane proteins is also quite a important challenge in the native MS world. We will hopefully get more of collaborations in the future. Now we're working with a lot of major pharmaceutical companies, and hopefully in the future we'll be able to bring that solution to you. Without actual data yet, we cannot say for sure, but we're looking at the mechanism of the enhancement and the software environment is generated. We're looking forward to see improve the analysis of the membrane proteins as well.
Yeah, I also can comment because membrane protein is a big interest for us. We're really working on it, but I can tell you, so that's why I show you exactly the proteasome example. The many people thinking they really low the capillary temperature to retain the non-covalent interactions, but what we find out specifically for UHMR is actually we never go the capillary temperature, we use minimum 250. We never go very low. However, what we do know, the S lenses has a much more bigger effect. If you want to keep the drug ligand or any kind of interactions, it's actually lowering the S lenses has a much bigger effect than lowering the temperature. This is how we also working with the membrane protein because we're working with the GPCRs. I know it's coming from the Carol Robinson group, which we had a huge, very big collaboration going on. And I exchanged the opinion, but that's what I recommended. And so the spray voltage, we were actually not going, we did try, and I think the lowest we can do 2 kV, which is latest we try in the latest source. It was actually not possible when it was just the emitter and the nano and then like our flex source. However, I don't think 1.1 it will be possible. I think 2 probably, but anyway, you guys, it's your call, right? But I don't, because we're not using the gases, and I think that's what we can probably find the conditions. So we only use this emitter so far in Lcms mode, but I think for the membrane proteins, it's probably you can do direct infusion. And if it's, and then you can spray, right? So the sensitivity is great. So far we're doing all this with a split because we cannot lower than 50 microliter per minute. We never run it, lower the flow rates, but I do think with the direct infusion, right? You can probably have a good results, yeah. But we didn't try it yet, so I should admit. So the membrane protein is on our to-do list, but we didn't try it yet.
Yeah. That makes sense, Rosa. Totally. I completely agree. And just to comment, for the voltage, spray voltage, so we do have a spec for spray voltage 0 to 6 kilovolts. So it is, like Rosa said, maybe she's not recommending it, but it is something we can apply and we can adjust and see if it works the best. But it's something that hopefully we can share with you guys in the future. The next question is that can you comment on giving some comment on the application the M3 emitter for glycoproteins. I know it is a very also very popular or interesting field. We have, like I've revealed in the application summary, we have done like say more of a benchmark testing of the HeLa digest and the plasma digest for discovery proteomics and just a proteomics setup comparison with both nanoflow method and with also in a high flow setting with comparison of a quantitative proteomics with comparison to high flow. Both show very much improved the sensitivity, and with a nano flow setting comparison, what you would see is a comparable sensitivity to nanoflow, but much better robustness and reproducibility in terms of the system robustness and the reproducibility of the peak area and the retention time. We have not really done a lot in terms of the glycoproteomics or glycoproteins. Hopefully, that's also something we can really apply the solution and this platform to help you address in the future. All right. Next question is, what is the droplet size? be some question that I can refer. I don't know if our Director of Engineering, Pen, are you online? Can answer that question. I know we the M3 emitter have a ID of 10 micrometers. So for the actual droplet size, I think what I can comment is that because we split the incoming microflow into nano spray, so the droplet size will be comparable to in the nano spray, nanoESI source. I don't know if that really answers your question, or if we have more comments from other Newomics colleagues here.
Okay, hi, Gaojin Wang here. Thank you for the question. So in terms of droplet, really depending on where do you see the droplet, at the beginning of the nozzle and also during a Colombo explosion, so the droplet start to decrease. When we talk about a droplet, if you say at the end of the nozzle tip, I think it's roughly the ID of the nozzle itself. But as the droplets start to form Colombo explosion, it becomes smaller and smaller. Right now, we have different size of ID for nozzles, from 5 to 10 to 20. So it really depends on the ID of the nozzle. Of course, also depending on the solvent conditions. Yeah. Right now, we have not really measured the droplet size. There's some way of doing this. Maybe in the future, we can use fluorescence imaging or some other technique to measure the droplet size. I hope this answers your question.
That's great. Good. Okay, so I have another question coming up here from Robert. Can clogging be a problem with ammonium acid mobile phase SUC applications? Rosa, let's hear a comment about it first through your experience, and I can also comment from here too.
We personally don't have any the clogging, but if you continue using the emitters, so but what you need to do, you really need to wash after you're done with your experiments, and if you're not planning to spray for like overnight, you really need to wash with water. But we also wash all our LC system if we finishing this this application, and then you need to probably run through some high organic, and then these nozzles can stay healthy. Yeah, but you need to take care of it. Yeah, and we're actually thinking about how we can implement, and we're talking to Newomics team about how to do more in automated way. Right, when we're done our experiments, it will be automatically washed with water first, right, and keep it stored. Yeah. That's a good question.
Yeah, it's actually a great question. Just to continue, we worked in the house collaboratively with the Rosa group, as well as, you guys have seen me showing some highlights of the Regenerance publication, which they actually evaluated different concentration level of ammonium acetate. And just like Rosa said, there is a potential clogging issue there, but because of the M3 emitter improved the sensitivity, it actually allows for different levels of splitting and different levels of flow rates to be used in order to best reduce that and to mitigate that. Another thing I think I want to mention is that all the emitters, they have a lifetime. So when you have single nozzle emitter, it will clog. With our multi-nozzle emitters, it could mitigate that clogging. But another thing is that your individual nozzle will probably gradually become clogged over extended usage. Right? I showed you the robustness data. It is because we have 8 nozzles here, 8 functional nozzles. It is unlikely to really interrupt that ongoing analysis until really a majority of the nozzle become clogged, and then that will take statistically a lot longer time. So this once again, will based on our experiments and the customer's experience, the multi-nozzle function really improves that robustness and to a certain degree reduce the probability of a clogging. Hope that answers your question. Yeah. Let's see. The next question, I think we have 2 more minutes, so, but we still have questions coming, which is really, really exciting. What is the optimal flow rate range with the M3 emitters in terms of the sensitivity and the reproducibility? I think that I can certainly comment. So we have the name of our platform, microflow nanospray ESI. So the optimal flow range obviously sits within the microflow range. So we have a specification. So the M3 emitter could work with the nano flow range. However, the best improvement of the performance you can observe is from the microflow range, and more specifically from 1 to 10 microliter per minute. So in that range, you can really see an enhanced sensitivity and reproducibility, precision, and robustness. To further comment on that, we're working hard in our R&D pipeline on different emitters, the next generation emitters that will work with really different flow rate range to be optimized for the different flow rate range and different applications. So there will be more things coming that fit your wider range of applications and flow rate needs. All right. All right, so this is probably the last question, and I'll refer again to Peng and Daojing. You guys can answer that. Is it possible to sonicate the emitter tip if they get clogged or dirty?
Okay, great question. We got asked by several customers. Typically, we wouldn't suggest you sonicate them because it's made of silicon. However, we did try in-house. It did work if we use a low power in sonication. So really depending on how you do the sonication and also how dirty it is or was the sample. We suggest actually use LC solvent to wash them using LC pump rather than doing the sonication because it's made of silicon. It is fragile, but it could withstand low power sonication. That was the experience, yeah. So was that the question about sonication to recover the performance?
Yeah, I think it's the question about to sonicate the tip if they get clogged or dirty. Yeah.
Yeah, yeah. The answer is we don't recommend it, but you could try with low power of the sonicator. Yeah.
Great. Thanks.
Okay.
Yeah, stop there. He likes the answer. That's great. All right. So our passed really fast, and we are very, very happy to today once again to present this to all of you and answer your questions. I know there's actually more questions coming along. We can certainly address them afterwards. We'll send you follow-up emails. And also, if you have more and more questions through the from the presentation, so you thought about it later on, think about it later on, feel free to email us to let us know. Once again, we thank you for your time and thank you very much for joining us today. We will follow up with a recorded video link of this WebEx, so you can actually, maybe refer to other people and share with others, and come back to us if you have interest on the MnESI platform and have more questions.
Thank you, everybody,
everyone,
for participating.
Yeah, thank you.
Thank you, Rosa.
Thank
great
you.
job. Okay, bye-bye.
Thank you. Bye.